When we talk about meiosis, it has some of the same steps as mitosis, but the first, it has two sets of cell division, and we're going to go through that. So again, meiosis reduces the chromosome number by duplication of these chromosomes, right? And then we have two divisions. So the first division, first, we talked about mitosis, right? And we just went from one 2N cell to two 2N cells, right? They were called daughter cells. They're identical. In this particular one, talking about is meiosis, and that's reductional, right? So the first, we're gonna have one cell that's diploid, and it's gonna go at the very end, it'll be four cells that are haploid. So here's another picture of those homologous chromosomes. Again, they have the same genes on it. They may have a different version of that gene. For example, if this right here was eye color, maybe dad has blue eyes and mom has brown. Okay. Or we could do hair color or whatever. Every parent has their own specific characteristics. So here we have our unduplicated chromosome or our monad. It's another name to call it. Okay, or we have our duplicated or our dyad. All right, so monad only has one copy. The dyads have two copies. We see that second copy again being made in synthesis. Talk about that part of a cell cycle. And like I said, the steps of meiosis is basically mitosis twice. In the first cell division of meiosis, so meiosis one, we're going to have our PMAT, prophase, metaphase, anaphase, telophase. We're going to reduce the number of chromosomes. This is where we split mom and dad's chromosomes up. And then meiosis two, we have to get rid of those sisters. All right. So here's prophase one. You can see the nuclear membranes breaking down. We have the homologous chromosomes finding each other. Right. So if chromosome one from dad finds chromosome one from mom, and all the other chromosomes follow the same uh, suit. And then when they're together, they can get kind of intertwined here. That location was called the synapsis, which is the pairing of the chromosomes. And then we're going to end up getting some mixture here. I think which move, which is not. I'm trying to get this thing on, but it's not. Right there. Okay, you can see how the chromosomes are all mixed up between mom and dad now from that crossing over. That's the first place in meiosis where we start to get genetic variation. So we're going to cross over. Here's our synapses where they're together, and then where it mixes up is called the chiasma, or if we talked about chimera, means a mixture. And this again is going to increase genetic variation. And metaphase one, they line up on that metaphase plate. We can see the spindles attaching to the kinetochores, and then anaphase is apart. If you remember in class, I said that was like the divorce. We split up the parents. And it's very important that we split them up. And then telophase one, we may start developing our nuclear membrane back and unwind the chromosomes, but it is short-lived because, we're going to go through cytokinesis and actually physically separate those. So now we have two diploid cells. The problem is they still have their sisters. So, and here's another picture. Oh, this is a sped up picture of prophase two. So again, we're going to get rid of that nuclear membrane and wind up if we started to unwind. Notice we line up on that metaphase plate, those two cells, single file, and here we split the sisters. Okay. All right. And then we're going to start with our telophase and cytokinesis to end up with four cells. Oop. So, um, I talked about this in class too, the difference between spermatogenesis and oogenesis, and how we have that spermagonian Once it goes through, we do end up with our four haploid cells. However, it is different in females. Right here, when we undergo our cytokinesis, it does not evenly split. Most of the materials go into one of those cells. The other becomes a polar body. And then when we undergo meiosis too, the polar body splits. And then we also, again, undergo uneven division of the cytoplasm content. So then we have another polar body and only one functional gamete, one functional egg. So with sperm, they end up going through meiosis. They have their four functional cells. Females only have one. And I talked about the female um, cycle here where, again, we have to go under mitosis so we don't get rid of all of our stem cells. And then one of the cells moves forward, one stays behind. And then each month during puberty, we're going to get one of these eggs ready, and they are rested. It stopped in prophase one. And then we have our cell division. We can see the follicle over here preparing that egg for ovulation. Once we get the go ahead for that particular oocyte to go to move forward for that woman's cycle, we're going to end up with right there our first polar body as we finish up meiosis one. All right, we do not finish meiosis two until the egg is actually fertilized. Once it's fertilized, then we'll go ahead and finish that up, and then you can see our three polar bodies in the one now fertilized egg. So here's a little side by side. So we can see our homologous pair. They're starting with two pairs of chromosomes. We can see with mitosis, we end up with two identical cells of what we started with, versus we have two cell divisions. Now notice right here in metaphase, okay, in mitosis, it's lined up single file. And in meiosis one, we're crossing each other for that partnership, right? They're all lined up. And then we undergo our second where we have to split the sisters. And then we have our, right here, we got to split the sisters. And then we have our four cells that are unique, genetically unique. Okay. Now, and again, this is showing here how they're lined up single file and here how they're partnered and the differences between those phases. Two identical cells for, so two diploid cells and four haploid cells. Now we talked about genetic variation. We talked about crossing over, but there is more than just crossing over. All right, so we get some genetic variation in two other places. Okay, prokaryotes only get genetic variation due to mutation when they um, mutate their DNA. They generally reproduce very quickly and they make mistakes. Okay. Now we have, I already talked about crossing over. You can see that again. We can see the mix up there. This chromosome's not all mom anymore, and this chromosome's not all dad. But we also have this thing called the law of independent assortment. Now, the law of independent assortment says that it doesn't matter how they line up on that metaphase plate as long as they're facing that partner. So you can see here, dad's on one side, mom's on the other. Here they have flipped, right? They're flipped. Like I said, it doesn't matter how they line up. And you can see how there are two possible ways here. And you can see how the offspring are different. We also have random fertilization, right? So numerous sperm go after that egg, and so we don't know which one will actually fertilize it, and each sperm is ideally a little different. And so that is the third type of location that we can get genetic variation. Sexual reproduction is very significant when we talk about evolution because it allows individuals within the population to be genetically different. We'll get to when you talk about evolution, we're not talking about evolution being that person, you or I or your friend, survives some kind of environmental change. We're looking at the population surviving. So having numerous individuals that do survive in order to keep our species going and surviving. And, those differences that those individuals may allow them to survive whatever has changed in our environment that has put individuals at risk. All right. So very, very important for genetic variation. All right. And that is the end of that presentation. I hope that was helpful for you, and I'll see you in the next video.